Okay, any questions? Tomorrow we're doing, I think it's our last regular staining exercise. We will do the PHB stain. That'll only involve a single slide. And you don't need a culture of any particular age or anything, so as long as you have some growth, you're good to go. So any questions about tomorrow? Don't forget tomorrow you will need to pull out your streak plates that you did and start evaluating them and seeing if you have different colonies. And you would want to sample those colonies and do gram stains so you can start kind of correlating and said, well, I saw a gram-positive cocci in that original culture that I did gram stain on, so I should be able to find a gram-positive colony and start, sorting that stuff out. So that's something we'll work on tomorrow too. As you can see from the last 5 years' stats, your exams are pretty much what I would have expected. Performance on that first exam has been fairly consistent over the years. And although the exams change from year to year, the basic difficulty of the exams is pretty much the same from year to year. So I'd just like to give you statistics to kind of see how you compare. I've got a question for you. It's been 2 years now that I haven't used a regularly assigned textbook. And one of the reasons that I stopped using the textbook was no one was reading it. Okay? You guys just don't like to read textbooks. And I couldn't see any reason to have to spend, $100 or more for a textbook if you weren't going to read it. So that's one of the reasons why I started posting the lecture notes, is I thought that the lecture notes were really going to kind of be a little kind of mini textbook. I wasn't, for the last exam, posting the lecture notes until after we covered the material, because what I was trying to prevent was printing those lecture notes out and then feeling like, number one, well, if I've got the lecture notes, I don't need to come to class. Or number two, coming to class and then just really not paying that much attention because I've got the lecture notes. On the other hand, for textbooks, the thing that really is the most beneficial for textbooks is if you can pre-read and read through the stuff before you come to class. So I was thinking about the other day when I was grading exams, and I thought, that would argue for you having the lecture notes ahead of time. So what do you think? Would it be helpful for you to have those lecture notes prior to when we start covering a topic? That way you could print them out, you could bring them to class, and then you could supplement those, because not everything I talk about in class is in the lecture notes, but then you wouldn't have to write down all the things that are in class already. So what do you think? Show of hands. How many of you would like to have the lecture notes available prior? Pretty much all of you. Okay? So I had started to post everything today and update it, so I will go back and I will post the material for the next topic, so you've got that ahead of time. If you can find the time to go through and just kind of look through the lecture notes prior to when we have a class, you're going to have a much better sense of kind of where we're going, kind of contextually what we're going to be talking about, and so I would really encourage you to use those. Okay? Any questions? Okay. So we're going to finish up microbial growth today, and then we will be shifting over to a new set of material on microbial metabolism. So when we talked about microbial growth last time, we were talking about some of the factors that influence the rate at which microorganisms can grow. And there were really two kinds of things that we talked about. Well, really three. We talked about some environmental factors that can influence the rate of growth. So things like pH, temperature, osmotic concentration, how much solute there is in the environment. When we're talking about nutritional factors, we're mainly talking about food resources available. So we talked a little bit about things like agar, broth, what they provide for the microorganisms, and what nutritional needs they provide. We introduced the concept of micronutrients at that time. And then in terms of gases, I said that the two primary gases we were going to talk about and concentrate on were oxygen and carbon dioxide. And we started out with oxygen, and I introduced this particular concept of an ROS. What does ROS stand for? Reactive oxygen species. And what about ROS? Is it good or bad? They can be harmful to cells, right? And particularly they are oxidizing agents, and they tend to remove electrons from other compounds and oxidize them. And that can create problems if the compounds that are oxidized are membrane lipids. That can affect the integrity of the membrane. It can influence the DNA and cause some pretty dramatic changes in DNA. There is some evidence that in humans that some of these reactive oxygen species are related to cancer, particularly those ROSs that have an effect on DNA. So what produces ROSs? We talked about two kind of major ways in which ROSs can be produced. What are they? No? Okay, as a result of aerobic respiration, which is good. We need aerobic respiration to occur. And in the electron transport system, normally what we expect to happen is that oxygen is going to be reduced to water. Oxygen is the final electron acceptor of those electrons passing through the electron transport chain. But sometimes that reduction that occurs is an incomplete reduction, and that can result in the formation of some of these reactive oxygen species. So they're a natural byproduct of an essential process, aerobic respiration. Where else can these be produced? Remember the other kind of category? Naturally in the atmosphere, particularly as light interacts with oxygen and can produce some of these reactive oxygen species. So one of the things we're going to be talking about and concentrating on when we talk about these more in a minute is that anytime oxygen is present and being used, it's likely that these things are going to be produced. And these are toxins. And they can be damaging to cells, including bacterial cells. So one of the things we're going to talk about is how does a bacterium cope with these particular kind of things if it's living in and using oxygen as part of its metabolism? So that's going to be kind of a theme that we're going to be interested in. Let's take a look at a couple of specific ROS types for just a second. Okay. One type of ROS species that's produced quite frequently is a compound called singlet oxygen. What happens in singlet oxygen is that an electron in oxygen gets boosted to a higher energy level, and that reaction, that oxygen then becomes much more reactive than it would be normally. And this, therefore, is a reactive oxygen species. This can happen during aerobic respiration, that singlet oxygen can be produced. Again, normally, we would think that this would be a bad thing. But as it turns out, there can be a situation where production of singlet oxygen can actually be beneficial. In important cells in your body, phagocytic cells, which are white blood cells, singlet oxygen is actually purposely produced. The cell intentionally produces it. And it does so within a lysosome. I don't remember what a lysosome is. Cell biology. What's a lysosome? Okay, it's a little membrane-bound vesicle, and one of the things that we talked about in cell was that it tends to be loaded up with digestive enzymes. And so when you bring things in from the outside, that those larger molecules or cells can be degraded into the molecules that they're made up of by these lipases and DNases and proteases. So one of the important things that happens in lysosomes is that intracellular digestion takes place. Well, phagocytic white blood cells are really an important part of your defense system. And one of the things that your white blood cells do is patrol around the blood or through your tissues. And if they encounter a bacterium, for example, they gobble it up. And they bring it into their cytoplasm by endocytosis. And therefore, that bacterium is enclosed within a membrane-bound vesicle in that cell cytoplasm. Now, does that automatically kill it? No. As a matter of fact now, that bacterial cell is protected inside the white blood cell from other parts of your defense system like antibodies. So that's kind of good for the bacteria now. But what happens? That particular vacuole that contains the bacteria can fuse with a lysosome, and now it's exposed to those digestive enzymes. And those digestive enzymes help to dismantle that bacteria and destroy it. And so that's part of the way in which phagocytic cells kill bacteria. They ingest them, and then they expose them to those digestive enzymes within the lysosome. There's another way that they kill bacterial cells too. And that is, within these lysosomes, a lot of singlet oxygen is produced. And for reasons that I'm not particularly aware of, the lysosome isn't negatively affected by the production of the singlet oxygen. But the bacteria are. And these reactive oxygen species that are being produced kill the bacteria. And then those enzymes can disassemble the bacteria into its constituent molecules. So one of the things that you can see within phagocytic blood cells is that when they ingest bacteria, there is a dramatic increase in oxygen uptake by those cells. That particular phenomena is called a respiratory burst. And basically, they're increasing their rate of aerobic cellular respiration. And they're pumping those reactive oxygen species into the lysosome to create this toxic environment that will kill the cells that they've engulfed. Okay? Medically, singlet oxygen has been used in a technique called photodynamic therapy. And this is a relatively new treatment that has been used to try to kill cancer cells. What they do in photodynamic therapy is they use monoclonal antibodies, which we'll talk about later on, but basically targeted guided missiles to deliver compounds to cancer cells specifically. They kind of paint the surface of cancer cells with these special compounds. And these special compounds are able to interact with light from a laser, for example. And when you shine a laser light on cells painted with these photodynamic compounds, singlet oxygen is produced. And therefore that specifically kills those particular cells. So only cells that are painted with the guided missile, the antibody, that contains that photodynamic compound are affected. So it's a very specific, targeted way to kill a particular type of cells. You just have to be able to develop an antibody that can deliver that compound to a particular cell, like a cancer cell. And that's pretty easy to do with monoclonal antibodies. So there's been an adaptation of this particular killing mechanism directed toward medical treatment of cancer. There's another important compound that's kind of at the root of other reactive oxygen species we're going to talk about that is also pretty important. And that particular compound is called the superoxide radical, or sometimes it's also called the superoxide anion. And this is produced during aerobic cellular respiration as well. It tells you here that this often begins with the auto-oxidation of a coenzyme, FAD. So during aerobic cellular respiration, this superoxide anion or radical is oftentimes produced. And it's harmful as well. So if you've got these superoxide anions floating around in the cell, that's problematic. They can oxidize membrane lipids, they can oxidize DNA. So some microorganisms have an enzyme called superoxide dismutase. And superoxide dismutase converts the superoxide anion into hydrogen peroxide. Well, that gets rid of the superoxide anion, but what is hydrogen peroxide? It's also a reactive oxygen species. Remember when we did the enzyme lab in cell biology? And we said one of the reasons that you have a peroxidase or a catalase enzyme is to detoxify and break down hydrogen peroxide. Hydrogen peroxide is just a less damaging version of a reactive oxygen species. So we're producing something that's not quite as toxic, but it's still problematic. Hydrogen peroxide itself can be converted into other reactive oxygen species, including the hydroxyl radical, by another enzymatic reaction. So you see how these superoxide anions are kind of the source of several other reactive oxygen species as well. Bacterial cells may or may not have one or more enzymes to deal with these reactive oxygen species, hydrogen peroxide. There's an enzyme called catalase, which breaks down hydrogen peroxide into water and oxygen. There's another enzyme called peroxidase, which breaks it down into water and oxidized NAD. So these enzymes, this one, and this one, are very important in the ability of microorganisms to tolerate these nasty things in yellow here, these reactive oxygen species. There are ways to get rid of those things. They're detoxifying mechanisms. So if you have the proper enzymes, like this one, or this one, or this one, then living in an oxygen-containing environment isn't a big deal. Because in the oxygen-containing environment, you are going to produce these things. But you can deal with them. You can break them down. But if you don't produce those enzymes, you don't have the right genes, you don't know how to make those enzymes, then being in an oxygen-containing environment, which means basically being in an ROS environment, is really a big deal. And typically it means you can't survive because you can't detoxify these byproducts, these nasty ROS species. So And that's going to be really significant. Now, there are some other ways in which microorganisms can deal with these reactive oxygen species. Besides having the proper enzymes, there's also a role that other compounds play called antioxidants. And I mentioned those last time. Antioxidants are compounds that kind of tie up or scavenge ROS species. And there are certain vitamins, for example, that are antioxidant compounds. So the ability to produce antioxidants is another protective mechanism. We also mentioned that there are some bacteria that can produce pigments, orange pigments, yellow pigments, those carotenoids, for example, which are scavengers for ROS species. So organisms that live in oxygen-containing environments have had to adapt and be able to evolve mechanisms to find ways to cope with the consequences of living in those oxygen-containing environments. And those consequences pretty much are the presence of those ROS species. Okay, so why are we doing this? Why are we talking about this? Well, in terms of oxygen availability, or probably even more accurately, oxygen tolerance, we can break microorganisms down into three categories. The first category we're going to talk about is going to be probably the most relevant to us in lab. Is that they use oxygen in their metabolism, and that they have one or more mechanisms that can detoxify those ROSs that oxygen is going to produce. Another category of microorganisms are those that don't use oxygen in their metabolism. So they would be happy living in an oxygen-free environment. They don't need oxygen. But they still do have mechanisms that allow them to detoxify the ROS species if they're present. Because remember, they can be naturally occurring. Third category, they don't use oxygen, and they don't have mechanisms available to detoxify any ROS species. How can you differentiate between these three classes of organisms? Well, there's a couple of ways we're going to do it in lab. And one is to use a device called a gas pack. Another is to use some growth media called thioglycolate tubes. So by inoculating plates and putting them in a gas pack, we can start to get some insight into these organisms' tolerance for oxygen and which category they might fall into. By inoculating them into tubes of thioglycolate broth, we can get similar information. And this information should give you the same results. It's just really two different techniques to get the same type of information. Okay, this is probably pretty confusing. Let's dig into this just a little bit more, and I think it'll become clear. So let's look at this first particular category. organisms that use oxygen in their metabolism and that have the ability to detoxify ROS species. One subgroup of that category are a group of microorganisms called obligate aerobes. Obligate means strict or absolute. These are organisms that absolutely require O2. If oxygen's not present, they die. But they have the ability to detoxify the ROS that aerobic metabolism typically generates. Another subcategory of being able to use oxygen and detoxify are a group of microorganisms called microaerophils. These are organisms that do require oxygen, but they require it in lower concentrations than normal. What's the approximate percentage of oxygen in the atmosphere? How much O2 is there in the air you breathe, do you know? About 20%. Okay. Most of it's nitrogen gas, N2, about 78% is nitrogen gas, then there's all the inert noble gases. But normal obligate aerobes require about a 20% O2 concentration. Microaerophils, somewhere between 2 and 10%. A third subcategory of this particular group are a group of microorganisms called capnophils. These also require lower than normal oxygen content. And these numbers are not important. Somewhere around 16%, okay, something less than the normal 20%. But additionally, they require elevated CO2. Anybody have any idea what the normal atmospheric concentration of CO2 is? It's pretty small. It's way less than 1%. It's like 0.03%. These require something like 4% for optimal growth. A fourth category are some really adaptable microorganisms that are called facultative anaerobes. Facultative anaerobes are just fine using oxygen if it's present and carrying out aerobic cellular respiration. But if oxygen isn't present, they have some alternatives. And there's some other ways in which they can survive and produce energy. So these particular organisms kind of can take it or leave it. They can use oxygen just fine, but they've got alternatives if oxygen isn't available. So they're kind of adaptable. it can be really important to be able to distinguish particularly between these two oxygen utilization patterns. Is your organism a strict aerobe or is it facultative? As a matter of fact, that oftentimes is an important dichotomy when you start keying things out in Berge's manual, especially the 7th edition, once you get down to that genus level and are trying to figure out species. So how do you do that? How do you determine whether something is facultative or an obligate aerobe? Well, here's one way you can do it. There is a device that will be described in a fair amount of detail in a lab in a couple of weeks called a gas pack. And what you can do is you can streak your organisms on regular old auger plates. You can put them in this plastic jar that has a very tight-fitting lid. And we're not going to get into the chemistry of this right now. We can talk about that when we do lab. But basically there are some chemicals in the form of a paper strip or a little envelope that you can put in there. And what they do basically is they create an anaerobic environment when you seal up that jar and close the lid. Basically what they do is they remove the oxygen by combining it with hydrogen to form water. Now normally these two particular things aren't very reactive. Oxygen won't just normally interact with water to form, with hydrogen to form water. So typically there's a catalyst that's involved as well that makes this reaction happen through temperature. But the final result is that this particular environment in here tends to become oxygen free and it becomes an anaerobic environment. So, if something is an obligate aerobe, should you get growth on those plates? No. Okay. What if it is facultative? Potentially, yes. Well, yes, it should. Because those organisms, if they're facultative, have other ways to respire and produce energy besides just strict aerobic respiration. They can carry out an alternative pathway called anaerobic respiration. And one of the things that I will want to stress to you later on is that anaerobic respiration and fermentation are not the same things. They're two different pathways. However, these organisms tend to grow more slowly. They give you lower growth in this anaerobic environment than they would if oxygen is present. So this anaerobic respiration is kind of a less efficient way of producing ATP. So, if I've got an experiment set up, and I have you streak some plates, and on those plates, you put your organism and you put them in the gas pack, and then we streak an identical set of plates and put them in a regular incubator where oxygen is present. If they grow in the incubator but don't grow in the gas pack, what are they? They're not obligate aerobes, are they? Because there's no oxygen. As it turns out, there's another category we're going to talk down here. They're going to be obligate anaerobes. What if they grow, but they grow better in the incubator than they do in the oxygen-free environment? Then they're facultative. They're limping along here and producing ATP by this alternative mechanism, anaerobic respiration. But it's not as efficient as normal aerobic respiration, so there's less growth. So these gas pack results are very, very important in terms of being able to make this particular determination. gas packs can't tell you whether something is a microaerofill or a capnofill. We're going to need some other particular tests to kind of tease that out. To determine if something is a microaerofill, typically what you need is you need results from thioglycolate tubes. Thioglycolate tubes are basically broth, but there is some chemical compounds added to the broth which tie up the oxygen and make it unavailable. And typically within these tubes, you will see different patterns of growth of organisms depending upon what oxygen environment they thrive in the best. Right up at the top, or the interface of the liquid broth, there is oxygen present. But as you get deeper and deeper into the tube, oxygen becomes less and less present. Okay? That compound ties up the oxygen in the media. But oxygen can diffuse from the air back into the media. But it's hard for the oxygen to diffuse through the liquid effectively. So this might have a little bit of oxygen, less, pretty much none. So by seeing the patterns of growth, you can then determine what type of organism it is. And this is something you will do in lab. So for example, this one right up here, look, all the growth is right up here at the top. What would that be? That would be an obligate aerobe. Okay? If you were a microaerofill, what would you tend to see? You would tend to see growth toward the top, but not exactly right at the top, and they don't show that pattern right here. There'd be a little band of growth in the tube. So if this is our aerobe right here, then a microaerofill might look like that. Okay, so deeper in the broth, this is where the surface level is. There's too much oxygen here, but now the oxygen content has been increased enough that it can grow. So looking for patterns of growth in thioglycolate tubes can give you important insights into the type of oxygen environment. There's a third device that we'll use in lab called a candle jar. And basically in a candle jar, you have a glass jar with a screw-on lid. And inside, you put a candle. You put your plates in here after they've been inoculated. And then you light the candle and you screw the lid on the jar. What's that going to do? Well, combustion of the paraffin in the candle is going to produce CO2. And that is going to elevate your CO2 concentration. What does combustion also do? Lowers the oxygen concentration, right? But it's not going to deplete it entirely like the gas pack does, because when the oxygen concentration gets around 16%, it's not enough to support combustion of the candle anymore, and the candle goes out. So basically, you put your plates in here, light the candle, screw on the lid, and let the candle burn out. And that generates this increased CO2, low O2 environment. You would want to do a comparable set of plates and put them in a regular incubator. And if they grew better in the candle jar than they grow in the regular incubator, that would be indicative of the organism being capnophilic. It's really important, though, to understand that the candle jar does not create an anaerobic environment. This is not the same as a gas pack. Okay? It's lower than normal O2, elevated CO2. In category number 2, there are a group of organisms that are said to be aerotolerant. And the word's pretty descriptive. They will tolerate the presence of oxygen. They don't use it, they don't need it, but they do have the necessary mechanisms to tolerate ROSs that oxygen typically generates. So they have one or more detoxifying mechanisms. That's not going to be an issue for us in lab, and we're not really going to need any tests to determine whether your microorganism is aerotolerant or not. That's kind of beyond the scope of anything we need to do. The last case of organisms are those that don't use oxygen in their metabolism and can't detoxify ROS species. Those are the obligate anaerobes. Naturally occurring obligate anaerobes live in environments like very deep in the mud, where oxygen has a difficulty diffusing into that particular environment. Interestingly, the inside of the gut, particularly the large intestine or the colon, tends to be fairly oxygen poor and almost anaerobic. So you have a very thriving population of obligate anaerobes living in your gut. What do these organisms do to produce energy? They can't use aerobic respiration. They're not facultatively anaerobic. Most obligate anaerobes use fermentation to survive. And fermentation produces gas. So it's those obligate anaerobes living in your colon that produces the gas that was produced in your colon. Practically speaking, how could we get an obligate anaerobe to grow? Either in a gas pack, or these researchers are working in a huge glove box area that is completely anaerobic inside. And all oxygen has been excluded, and therefore these organisms can grow, because otherwise ROS species associated with presence of oxygen would be lethal to them. Okay? So in these last categories, in these obligate anaerobes, oxygen is a poison. And it's not oxygen really that's the poison, it's the ROS species that the oxygen produces. Yes. So are any of your unknowns going to be obligate anaerobes? Obviously not. They grew in the incubator, right? And there's oxygen in the atmosphere. So really, the big determination that we're going to have to make in terms of identification of your unknown is your organism an obligate anaerobe, or is it facultative, or is it a captive? And so in lab, we will do a gas pack, which will help to give us information about both this and this. We will do the thioglycolate tubes, which will give us also information about this and this. And it's always nice to have corroborating information about your unknown. If you get the same answer from 2 different tests or 2 different sources, you're much more confident. But then we will also do this gas pack, the candle jar, to determine if your organism is capnophilic. So oxygen is really important. So we've introduced some terms now that probably you're going to need some refreshment on, and some concepts that you probably haven't heard of before, particularly this idea of being facultatively anaerobic, and what anaerobic respiration is, and how it differs from aerobic respiration, and how it also differs from fermentation. So we're going to do some review of metabolism, talk about some things that you should remember from cell biology, but then add some things that microorganisms can do metabolically that human cells can't. Okay? So that'll be the next topic. So I've already posted the PowerPoint slides and study guides for this next section, and I'll go ahead and post lecture notes for this next section as well.
One of them last night, I was looking at it.